Hello everyone, my name is Deepshikha Sharma, a student of Chandigarh University pursuing BSc Biotechnology and currently in my third year. Today I will be presenting my summer internship project on exploring the transcriptome and the understanding gene expression dynamics in breast cancer progression. This project was completed during my 45-day internship at OmicsLogic in Bioinformatics and Data Science India Private Limited under the guidance of my mentor, Mr. Harsh Kumar. Through this presentation, I'll take you step by step into how I explored transcriptome data, analyzed gene expression, and understood the progression of breast cancer at the molecular level. So, before going further, let's discuss what exactly is breast cancer. So, breast cancer happens when cells in the breast starts growing abnormally and uncontrollably, forming a lump or a tumor. These tumors can invade nearby tissues or spread to other parts of the body. According to World Health Organization in 2022, approximately 2.3 million women were diagnosed with breast cancer, and sadly 670,000 women passed away. This increase in the breast cancer is alarmingly increased by about 3% every year. It may be due to lifestyle choices and aging population. So, how do doctors diagnose breast cancer? First, they run a physical examination, such as to detect lumps, then imaging tests like mammograms, ultrasounds, or MRIs, and finally, the most confirmatory test are biopsy, where a piece of tissue is studied under a microscope and checked for cancer cells. Then certain follow-up tests are also run, like additional tests to determine if cancer has spread. So in this image, you can see different types of breast cancer classified based on whether where they originate. Or for example, some begin in the milk ducts, which are called ductal carcinoma or ductal cancer, and some in lobules, which are called lobular carcinoma or lobular cancer. So, here below you can see the normal cells of the duct wall and then which are transitioning through the benign stage to go into the atypical hyperplasia stage where abnormal cells growth can be seen. Then these black cells can be called as cancer cells and these are inside the duct wall. Then when these cancer cells break through duct wall, these are called invasive cancer. And when cancer cells travel to other parts of the body, either through blood streams or through lymph vessels, they are called metastatic. This gives rise to the malignant property of the cancer. That means they can travel to other body parts and can cause tumors or cancers there. So now, what was the problem and what was the purpose of my study? So, the problem with breast cancer research is that while we know it's one of the top causes of cancer related deaths, we don't fully understand the molecular changes happening inside cells as the disease progresses. There are certain conventional methods which are to detect cancer, but often miss the dynamic changes in the gene expression. That is how certain genes get turned on or off during cancer progression. In this statistical study also from the Canadian Cancer Society, we can see the percentage of all estimated cancer deaths in women in 2004, where 87% of the deaths were due to other cancer types such as lung cancer or cancer of any other body part and breast cancer occupied about 13% of the total statistical study, which means that breast cancer is still a significant cause of cancer in many women around the globe. So, here in my study, I explored the various biological pathways and gene expression, mainly to identify differentially expressed genes, which are called DVGs, which show the significant changes between normal and cancer cells. then to uncover pathways and biomarkers that could improve early detection and targeted therapy. So, what were the findings from my original paper? The paper which I used for the study contained 12 samples of the cells which exhibited different stages of malignancy, including samples from normal or non-tumorigenic cells for comparison. The study analyzed 12 samples, which were divided into four categories. First was MCF10A, which contained normal breast cells, or we can also say non-tumorigenic cells. Then MCF new T, which were for benign hyperplasia, which means non-cancerous cells overgrowth. And then we move to MCF10-1, which contained for cells for atypical hyperplasia, which is a precancerous condition. And then to MCFCA1, which are for invasive cancer cells. So, by studying these all 12 samples, we can study the potential biomarkers which can be used for the early detection and therapeutic targeting, and can also baseline the transcriptomic analysis and sequences for a particular cancer. So here is the methodology or the study pipeline which I followed during my study. First, I collected or retrieved the RNA sequencing data from GEO with the accession number of GSE241432, which contained the 12 samples as I already explained. Then I moved to exploratory analysis where I performed PCA or principal component analysis. PCA, which is used for simplifying the large data and shows how samples grouped together. Then I moved to differential expression analysis using GEO2R for each separate condition, where I identified which genes were significantly up or down regulated. Then I moved to functional enrichment analysis, where I used ShinyGo and KEGG for exploring what biological processes and pathways these genes belong to. And then finally, I compared across all the three conditions with the Vani 2.0 and found out overlapping genes between different stages. So let's move to the results section for exploratory analysis, which means PC analysis. The PCA plot shows here a clustering of samples based on gene expression. Each dot represents a sample, and the distance between them indicates similarity or difference. So there are samples for normal cells, cancer cells, benign hyperplasia, and atypical hyperplasia shown in different colors. We can see that normal and benign hyperplasia cells are clustered closely, and same goes for atypical hyperplasia and invasive cancer cells, which are grouped closely. Here, clustering means that the cells or their functions are closely related, not overlapping each other, but they are significantly related to each other. Or we can say here that atypical hyperplasia can be a transition from healthy to malignant stage of cancer. Here, this clustering helps identifying biomarkers of malignancy, which can be used for further cancer analysis. As we move to the differential gene expression analysis, these in these plots we can see that there are red dots which are for upregulated genes. There are blue dots which are for downregulated genes, and there are also gray dots here at the base in the center, which are for the genes which have no significant change during the transition of normal cells to cancerous or malignant cells. Um, these volcano plots shows us how the genes affect a particular condition and what genes specifically are related to a particular condition. Here, the number of significant genes or significant DEGs, which play a very important role in the transition of normal cells to cancer cells were found. In benign hyperplasia versus normal condition, these were found around 2626. In atypical hyperplasia versus normal condition, these were around 1396. And in cancer versus normal condition, these were around 2586. Um, so these plots which have shown here, in here the vertical axis shows statistical significance and the horizontal axis shows fold change in gene expression. And the spread of red and blue dots represent the extent of gene alterations across stages. Here is also a Venn diagram generated using Venn 2.0, which illustrates the number of common DEGs in all the three conditions. The conditions here are mentioned: benign hyperplasia versus normal, atypical hyperplasia versus normal, and cancer versus normal. So, in all the three conditions, from all the genes which are involved in all the three conditions, 535 genes were found common. It means they have significant role in each of the conditions, whether upregulation or downregulation of the particular gene. Let's move to the pathway enrichment analysis. After identifying DGs, I move to this analysis using KEGG and gene ontology. KEGG pathways highlights which cellular signaling routes are disrupted, and GOTOMs classify gene functions into three major categories, which are biological processes like cell growth or apoptosis, molecular functions like enzyme activity, and cellular components like the nucleus or membrane. So these plots which are shown here demonstrates the cake enrichment pathway for benign hyperplasia versus normal condition and for atypical hyperplasia versus normal condition. Here is also for the invasive cancer versus normal condition, which is shown here. If we see in the plots, there are many pathways which play important role in progression of a particular disease, and such as the pathway for malaria, the pathway for ECM receptor introduction, and the cell adhesion molecule pathway. These all play a very significant role in the progression of benign condition of the cancer. Similarly, in atypical hyperplasia condition, there are many pathways which are upregulated or which are downregulated, but but play a significant role in the progression of the disease. There are also many pathways such as interleukin 17 signaling pathway, chemical carcinogenesis, DNA adapts pathway, amyloidosis pathway, which play a very significant role in the progression of invasive cancer, especially. So, among all these pathways, there are many pathways which are also common in all the three conditions, which we will go later on. These are the pathway signatures. It means that there are certain pathways which are responsible for the progressiveness of the tumor. There are certain pathways which are responsible for the suppressiveness of the tumor. And there are many common pathways which I already explained in all the three conditions. So, the tumor progressive pathways are responsible for proliferation, invasion, angiogenesis, and survival and overall cancer aggressiveness of the disease, such as interleukin 17 signaling pathway, ECM receptor interaction, focal adhesion, cytokine receptor interaction, etc. These pathways are involved in the progression of the tumor, and these pathways listed here are responsible for the suppression of the tumor. They are downregulated or altered in cancer, which are also these pathways are also responsible for the progression of the tumor by suppressing certain other pathways or genes, and such as TGF beta signaling pathway, drug metabolism pathway. These are suppressed, and by due to the suppression of these pathway, cancer progression goes further. And then there are certain common pathways which are included in all the three conditions. It means they are highly significant in all the three conditions. Then there are these gene ontology enrichment analysis, which shows that there are certain number of genes, and the genes which are responsible for certain functions among biological process, molecular function, and cellular component. These plots are for the benign hyperplasia versus normal cells. In these plots, we can see that there are, for example, 487 genes which are responsible for the regulation of multicellular organismal process. And if these genes are upregulated or downregulated in a particular condition, invasive cancer condition can arise. Similarly, in molecular function, there are protein binding, signaling receptor binding. All these functions are done through a certain number of genes, and if there is any abnormality or alteration in those genes, invasive cancer condition can arise. These are for the atypical hyperplasia versus normal condition, what genes and how many number of genes are responsible for what functions, and what does the alteration of these functions can lead to in all the three conditions: biological process, molecular function, and cellular component. Then we arrive to the discussion part of the presentation. That from all these analysis, here's what I learned. Distinct gene expression profiles access study each stage of malignancy. Atypical hyperplasia clusters near invasive cancer, confirming its role as a transitional stage. There are certain genes like CEACAM6, AGR2, CLC6A14 stand out as biomarkers, which means that these genes play highly significant role in the upregulation of these genes. It means that these genes, when upregulated, can cause cancer, and in this way, they can also use as biomarkers or therapeutic targets. By targeting these genes, we can suppress the invading cancer. Then, instead of random disruptions, cancer progression involves targeted specific pathways. This means transcriptomic analysis can truly improve early diagnosis, personalized treatment, and therapeutic strategies in breast cancer by knowing what genes and what are their functions which are upregulated or downregulated in a particular stage of malignancy. To conclude, using PCA, GEO2R, Veni 2.0, and ShinyGo identified significant differentially expressed genes and pathways. 535 common DGs were found across all stages of malignancy. Key genes like CEACM6, AGR2, CLC6A14 may serve as strong biomarkers. This project not only deepened my understanding of breast cancer genomics, but this also enhanced my practical skills in transcriptomic data analysis and bioinformatics tools. Here are the references which I used for my study and gaining the knowledge about breast cancer. And so this was all about my presentation. Thank you so much for your attention.